Welcome back to Genetics Part 2. I'm sure you were anxiously awaiting the start of this video because you're so excited to see what happens here in this dihybrid cross. This freckles, no widow's peak, no freckles, widow's peak, and then last, no freckles, nor widow's peak. So our phenotypic ratio of 9 to 3 to 1, 3 to 3 to 1, we've got 9, 3, 3, 1, right? So all things being equal, 9 times out of 16, you'll have a child with freckles and a widow's peak. 3 out of 16, freckles, no widow's peak. 3 out of 16, no freckles, widow's peak, and 1 out of 16 times neither freckles nor a widow's peak. So a little bit more than half the time, their offspring will have both freckles and a widow's peak. Exciting. Using flies to do this is pretty fun because then you can put them under the microscope and see what traits the offspring have. It's pretty cool. And then another really interesting thing to do, which in a general biology class I would have students do as an assignment, is a pedigree. And in the pedigree you get to see at least 3 generations of what's going on with different traits. And so you usually do circles for females, squares for males, and then fill in for the affected ones. So let's say tongue rolling, right? Versus no tongue rolling. And then you could see with a bigger picture a pattern. And so here, there's a non-tongue roller male and a tongue rolling female. And now if you see here, look, they had 6 kids. 1, 2, 3, 4, 5, 6. 5 boys and 1 female. And so because they had kids without the tongue rolling trait, I know that that person has to be able to send on a little t, right? And then these kids obviously got a little t, so they're all big T, little t, big T, little t, little, little, little. And then this person comes in, so his partner, and they have 2 little t's and 1 that has to be heterozygous. And then here, little t, big T, little t. So based on the pattern, I could identify the genotypes of all individuals. Now, it won't always work. I mean, what if they only had those first 3 kids? Then you might think that it's big T, big T. You couldn't tell, right? You wouldn't know for sure. That's pretty neat. Okay, if we're looking at like diseases that are oftentimes recessive, where you need 2 bad copies of the gene to get the disease, people who are heterozygous can be called carriers. So let's say this is for cystic fibrosis, right? 1 bad chlorine channel gene, you're okay. But you have, so let's do chlorine channels, or cystic, big C, little c, right? So this mom doesn't have cystic fibrosis, but she's a carrier. Same for this dad. So now, in their offspring, one of those kids is going to have the trait. 2 are going to be carriers, and 1 is not going to be a carrier. Cystic fibrosis is looked at a lot because it's the most common inherited gene among white people of European descent. So they wouldn't know they were carriers unless it's been in their family or they got genetic testing. Right? Exciting. Now let's look at some more principles of how things overlap. Okay, so what we've been talking about before was complete dominance. 1 trait, 1 allele totally masks the other. Dominant, masks, recessive. So in codominance, they're both visible. Like This one, the best example is blood groups, ABO. If you have the A protein, A's out there. If you have the B, B's out there. If you have both, A and B are both out there. They don't mask each other, they're just both there. Whereas incomplete dominance, they both are there, but they're not as strong. So like hazel eyes is a good example of this. In plants, a fun one is red gene and white gene makes the flowers pink. Cool. Okay, so what we've been looking at is monogenic, one gene. But most of our traits are polygenic. They have many genes that code for them. So skin color is a good one. There's 6 genes for that. Height also. And what you'll see there is that like this for height shows all the tall version, all the short, and most of us are somewhere in the middle, this like bell curve. And so here's men standing out at different heights. Some very short are somewhere in the middle. So genes on the same chromosome near each other are considered linked because they're likely to go together and they won't get separated during meiosis. So we look at that a lot with flies. And so if they're not linked and you did one of those cross comparisons, you would expect equal odds of getting any of those types. Whereas if they're linked and you cross them, you're far more likely to see like the black coloration with these vestigial wings and the gray with the big wings and vice versa. It's more likely that the linked ones go together and that they don't switch. Okay. Cool. And then if traits are on the X or Y gene, they're called sex-linked. And the X chromosome is much bigger than the Y chromosome, so it's far more likely to be X-linked. Right? And we've talked about, I think, all of these before. Red-green color blindness, hemophilia with clotting, Duchenne muscular dystrophy, where your muscles aren't as strong. And so this is showing how that works because X-linked, the male only has 1 chance at a working gene, which here they've shown in orange, whereas females have 2 chances to get a working gene. So it's far more likely to see these disorders in males than females. For a female to be red-green colorblind, her father has to pass on a mutated X and her mother has to pass on a mutated X, which means her father has to be colorblind and her mother has to be at least a carrier. Right? So this is showing basically their Punnett square, and down here is the same thing. Unaffected, carrier daughter, affected son. Right? If mom passes on the disease X, the daughter will get a normal one from the dad, but the male only has 1 chance. And there's the Punnett square for it. And then a really fun thing is sex-influenced genes, where you have to have the genetics and also hormones. So an example is male pattern baldness or frank baldness, so that's Frank from It's Always Sunny. So there you need the genes for balding and high levels of testosterone. So that's exciting. Okay. And that is the end of this brief tour of genetic inheritance. Have a lovely day.